Hello everybody, welcome to online lecture in Rao Bahadur Narayan Rao Borage College, Srirampur. Today we are going to study nucleic acid. Nucleic acids such as DNA and RNA. DNA and RNA. DNA. Chromosome contain DNA. Chromosome contain DNA. It is also called as deoxyribonucleic acid. DNA is composed of sugar molecule, It is pentose sugar. DNA is composed of sugar molecule. It is pentose sugar. Then phosphoric acid or phosphate group. And nitrogenous bases. Nitrogenous bases. So here nitrogenous bases are mainly of two types, that is purines and pyrimidines. Purine and pyrimidine. Purines are the adenine guanine, and guanine, while pyrimidines are cytosine and thymine. So once again, DNA, they are present in chromosome. So DNA is also called as deoxyribonucleic acid. It is made up of sugar molecule, especially pentose sugar, then phosphate group and nitrogenous bases. Nitrogenous bases are of two types, that is purines and pyrimidines. Purines like adenine and guanine, cytosine and thymine. So adenine, guanine, cytosine and thymine. So that these nitrogenous bases, which are always forming a pair like that, adenine is always paired with thymine, guanine always paired with cytosine. Adenine is always paired with thymine, guanine is also paired with cytosine. DNA, it is nothing but a long chain made up of alternate sugar and phosphate group, and the sugar is always deoxyribose sugar and it always join with phosphate group. So DNA, it is a double helical structure. It is a double helical structure. It is made up of pentose sugar, that is deoxyribose sugar, then phosphate group and nitrogenous bases. So that nitrogenous base, which are just like a step of a stair or staircase, आपली स्टेअरकेस असते किंवा जिना असतो बघा, बऱ्याच वेळेस असा ट्विस्टेड जिना असतो. अशा प्रकारचा ट्विस्टेड जो जिना आहे तर त्या जिन्यासारख्या याच्या त्या स्टेप आहेत आणि त्या प्रत्येक स्टेपमध्ये त्या ठिकाणी आपल्याला पाहायला मिळणार आहे की पेंटोज शुगर, फॉस्फेट ग्रुप आणि नायट्रोजनस बेस यांची त्या ठिकाणी अशी पेरिंग झालेली आपल्याला पाहायला मिळणार आहे. मग आता इथं हे जे स्ट्रक्चर हे असं डबल हेलिकल स्ट्रक्चर किंवा असं ट्विस्ट आहे आणि हे ट्विस्ट झालेले स्ट्रक्चर जे आहेत त्यामध्ये त्यांच्या ज्या पेरिंग आहेत, पेरिंग अशा विशिष्ट पद्धतीने पाहिलं, ज्याला आपण ॲडेनाइन इज ऑलवेज पेअर विथ थायमिन, ग्वानिन इज ऑलवेज पेअर विथ सायटोसिन. इन दॅट double helical structure there is one thread is downward or lagging and another which is upward, that is leading and lagging, leading and lagging. त्यातला एक जो थ्रेड आहे तर तो वरच्या दिशेने जाणं त्याला लिडिंग म्हटलं आहे, दुसरं खालच्या दिशेने जाणं त्याला लॅगिंग थ्रेड असं म्हटलं आहे. अशा प्रकारचा हा DNA जो मॉलिक्यूल आहे, हा मॉलिक्यूल कसा आहे तो डबल हेलिकल स्ट्रक्चर आहे. इट इज मेड अप ऑफ पेंटोज शुगर, फॉस्फेट ग्रुप अँड नायट्रोजनस बेसिस. In case of a single strand of DNA, it consists of several or thousands of nucleotides one above the other. Out of that, one strand is 5' 3' and another which is a 5' 3' 1. These are the opposite to each other and they are placed just above it. In case of RNA molecule, the cytosine is replaced by uracil. Cytosine is replaced by uracil. It is a single stranded. Then RNA, it is found in most of the organism and they are having with a hereditary material, which is a hereditary material, and generally RNA it is a site of protein synthesis. RNA. It is also called as ribonucleic acid. In RNA, the cytosine which is replaced by uracil. RNA, it is a site of protein synthesis. It is site of protein synthesis. It is of three different types: mRNA, rRNA and tRNA. mRNA, rRNA and tRNA. RNA are of three different types, that is mRNA, rRNA and tRNA. So very first mRNA, they are called as messenger RNA. mRNA, they are called as messenger RNA. mRNA are called as messenger RNA. They are mainly responsible for carries genetic information. That information is mainly for arranging the amino acid in definite sequence. Arranging amino acids in definite sequence. In the account of cellular RNA, it may be 3%, and the genetic information from a complete polypeptide chain is called as cistron, and that cistron play an important role in synthesis of proteins. Then rRNA, which is also called as ribosomal RNA. Ribosomal RNA. Near about 50 to 60% of ribosomes are rRNA. It is synthesized in nucleus, and it also responsible for protein synthesis. The next, that is tRNA, which is called as transfer RNA. tRNA molecules are smaller, consisting near about 70 to 80 nucleotides. It is single-stranded, which are mainly responsible for transmission of message. For particular amino acids in the process of protein synthesis. The tRNA, which are showing a structure which is just like a clover leaf. Clover leaf. the chlorolip model. It is just like a claw, or मराठीत त्याला आपण लवंग म्हणतो, त्याच्यासारखं त्याचं स्ट्रक्चर आहे, म्हणून त्याला क्लोरलिप मॉडेल असं म्हटलेलं आहे. दॅट क्लोरलिप मॉडेल इट कन्सिस्ट 4 different parts, that is DHU, then amino acid binding site, anticodon loop, and ribosome regeneration. In the anticodon loop of tRNA, 3 unpaired nucleotides are present, which are called as anticodon, and which pair with codon present on the mRNA, so that the specific amino acids is attached at the 3 end and in acceptor stem of the chlorolip of the tRNA, so that these nucleic acids, that is DNA and RNA, play an important role in the transmission of genetic information. So according to the scientists Watson and Crick, the DNA molecule consists 2 strands or twisted strand, that is each site having a end, that is 3' 5' end. Each side has a 3' 5' end. Out of that, one is leading and another is lagging side, and each DNA molecule is paired with a pentose sugar, then nitrogenous base, and there is also a presence of nitrogenous bases, that is always paired with adenine, is always paired with thymine, guanine is always paired with cytosine. So the DNA molecule which play an important role in formation of chromosomes, and chromosomes which are the components which carries the genetic information from one generation to next. Then enzymes. Enzymes are essential for different chemical reactions takes place automatically or at the given time in living cell. It requires for chemical reaction in body, which act automatically at a given time in a cell. The reaction takes place at body temperature. Reaction takes place at body temperature. So that for the normal reaction of enzyme, there is normal temperature is required. So normal body temperature is 37 degrees centigrade. If it is increase or decrease, the reaction is not completed or it is not showing its proper effect. So that proper temperature and pH is required for the activity of each enzyme. So each enzyme catalyzes a small number of reaction, and especially they are specific for specific activity. Each enzyme which are having a specific activity. So that enzymes synthesized by living cells retain their catalytic property even when extracted from the cells. So that enzymes play an important role in the each and every reaction. So nature of enzyme is also important. On the basis of chemical composition, enzymes can be into two categories. Number one, which is a purely proteinous enzyme and conjugated enzyme. Purely proteinous enzyme. And second, which is conjugated enzyme. Conjugated enzyme. Purely proteinous enzymes, such as protease enzyme, that responsible to splitting the proteins. Proteases enzymes, which are responsible for splitting or breakdown of proteins, or they are breakdown into amino acids. Then conjugated enzymes are made up of protein to which a non-protein prosthetic group is attached. Enzymes which require certain organic compound and inorganic ions for their proper activity. Then properties of the enzyme. Properties of the enzymes. All enzymes are protein. All enzymes are protein, but all proteins are not enzyme. All enzymes are basically made of protein, but all proteins are not enzymes. They are act as a catalytic agent and influence the speed of chemical reaction. आता हे एन्झाईम्स हे कॅटलिक म्हणून त्या ठिकाणी आहे आणि ते त्या ठिकाणी त्या रिएक्शनचा त्या ठिकाणी स्पीड वाढवण्याच्या दृष्टीने त्या ठिकाणी उपयोग त्या ठिकाणी होऊ शकतो. अँड a small quantity of enzymes is required for the catalytic reaction, and each enzyme is specific in action. Then the enzymes, they are also showing a reversible reaction. So enzymes are very sensitive to temperature and pH. If the temperature is decrease or increase, or pH is decrease or increase, it shows abnormal activities and the reaction is not completed.